examples of sex-linked inheritance. The first example is color blindness. Color blindness, it is caused due to recessive X-linked genes which prevents formation of color sensitive cells, the cones in retina of eye, due to which the person is unable to distinguish between these two colors, that is red and green color, and both the colors appear grey to him. We'll study this with the help of a cross. So to proceed with the cross, we must know the genotypes that are used for these. As I have already said that this type of inheritance is crisscross inheritance. That is, it is passed from parents to their offsprings, that is from grandparent to grandson via daughter. So in the first cross, the parents that we are choosing is color-blind male and normal female. That means when marriage takes place between a color-blind male and a normal female, what will be their offsprings? So for that, we must know the genotypes for the parents. Now this is the genotype chart for color blindness. This is for male and this is for female. The normal male, generally males have different chromosomes, that is one X and one Y chromosome, whereas females, they carry same chromosomes, that is both the chromosomes are X chromosome in females. Now as we have said, this is X-linked genes, so genes will be present on the X chromosome only. So a normal male will have genotype as X, on which the gene is present, which is represented as capital C, and this is normal Y chromosome. In normal female, this is X chromosome, both are X chromosomes, and on both the X chromosomes capital C is present, that is gene for color blindness is present. Color blind male, it will have the gene on X chromosome, but this is small c, and this is normal Y chromosome. Whereas color blind female, it will have genes on both the chromosomes, and these are recessive genes, whereas these are dominant genes. So this is recessive gene for colorblind male. These are recessive genes for colorblind female. Generally, males, these are not carriers. Females are carriers and they carry one dominant gene and one recessive gene. So this is the genotypic chart for color blindness. The most important thing that you must take care is the difference should be clear cut between the capital and the small alphabet, because if the teacher or the examiners, they are not able to distinguish between capital C and small c, you will come in trouble. You won't get the marks for that. Hence, clearly write the capital alphabet and the small alphabet wherever required. Now let's place this genotype in the given cross. So now we'll proceed with the cross. Now as we have said, the parent, one is colorblind male and the female is normal female. So for colorblind male, what is the genotype for colorblind male? It is X, small c and Y. X, small c and Y. This is the genotype for colorblind male. For normal female, both the X chromosomes, they will carry dominant gene. So these are capital and this is small alphabet. Now the next step is gamete formation. Since it is heterozygous, two different types of gametes will be formed. So the first one will be X. This is small c that we have to remember, and this is Y chromosome. Both are same. This X is having capital alphabet. This X is also having capital alphabet. So both of these are capital, that means they are dominant. Then comes a F1 generation. So for F1 generation, fusion of male gamete and female gamete is required. So one gamete has to be from male parent and the other gamete has to be from female parent. So this first we'll have to cross with this one and then with this one. Similarly, Y chromosome we have to cross with this one and this one. So let's proceed with that. This is one of the male gamete. This we are crossing with the female gamete. So this is X and small c. This is X and capital C. So first we'll write the capital one. This is capital and this is small one. Now the same gamete, this we have to cross with other one, that is female gamete. This is male gamete and this is other female gamete. So this is other female gamete. This is X and capital C. This is X and small c. But we have to write the capital one first. So this is X, capital C. This is X and small c. Capital C, small c. Clearly it has to be differentiated. Then we have to cross the next one, that is Y chromosome. So this is Y chromosome. This will have to cross first with this one. Even though Y chromosome comes first, still we have to write the X chromosome first. So first we'll write X, capital C and Y. Again this chromosome will have to cross with the female gamete. So it will be, again, even though Y is coming first, still we'll have to write this one first. So it will become X, capital C and Y chromosome. In this case, both the chromosomes, both the offsprings, they are heterozygous. That means one is dominant, one is recessive. In this case also one is dominant, one is recessive. In both the offsprings, X is carrying dominant genes. Hence, the offsprings that will be produced will be, these are carrier females. Carrier females or carrier daughters we can see. Generally the offsprings, they are called as daughters and sons. Whereas these two offsprings, these are normal sons, because they are not carrying the recessive gene on X chromosome. That means 50% offsprings, that is daughters, they are carriers, whereas 50% offsprings, that is sons, these are normal. Now the gene has been transmitted from father to daughter. So this is the first half of the cross. For the second half, what we will have to do? We will have to take the parents as carrier daughter. When she grows up and she marries to a normal male, the genotype for this will be, just now we have said that the carriers will have one capital and one small one. And normal male, it will have the dominant gene and Y chromosome. So these are the genotypes for the parents. Now comes gamete formation. Gamete formation will be, this will be X and capital C, this will be X and small c. Whereas the gametes for male will be, it will be X, capital C and Y chromosome. So these are the parents for next half of the cross. From father, the daughter has received the genes for color blindness. She is carrier, that means she is carrying the gene, but she is normal, that means she is not suffering from the color blindness disorder. Now she is married to a normal male and these are the gametes. So now gametes, we have to cross them. The cross will be like this. This is the first offspring that is produced. X capital C, X capital C, X capital C, X capital C. What does it represent? It represents that this is normal daughter. Why daughter? Because both the chromosomes are X chromosomes, hence the offspring will be a daughter. Now we have to cross the other one, that is this gamete. We have to cross with the other one, this one. Now see, this is X capital C and this is Y chromosome. X capital C and Y chromosome. When the gene is dominant and when the chromosomes are, one is X and one is Y, the offspring will be a son. So this is normal son. Then comes the next cross. This is female gamete and this is male gamete. So this is X capital C, this is X small c. First we have to write the capital one and then we have to write the small one. So X capital C and small c, they represent what? They represent as carrier daughter. Then the last one. This is one gamete. This is other gamete. It is X small c and the next chromosome is Y chromosome. Since both the chromosomes are different, that is one is X and one is Y, the offspring that is produced, it will be a son, but this is a recessive gene and it shows that it is color blind son. So from this cross, one thing is clear, that the genes from father are transmitted to the daughter, they become carrier, and when they are married to a normal person, it is transmitted to the carrier daughter's son, due to which the son becomes colorblind. So 4 offsprings are produced, in which one is normal daughter, one is normal son, one is carrier daughter and one is colorblind son. For carrier daughter, one has to be recessive gene, other has to be dominant gene. But since there is no alternative allele on the Y chromosome, the son, it becomes colorblind. Females can only become colorblind when the recessive gene is present on both the X chromosome, then only female can become colorblind. This is the second example of X-linked inheritance, and the example is hemophilia. Hemophilia, it is also called as bleeder's disease. Hemophilia is an X-linked recessive disorder in which the blood fails to clot or the clotting is very slow. Now let's see this example with the help of a cross. Again, this is also crisscross inheritance, in which the genes will be transmitted from father to daughter, and from daughter it will be transmitted to her son. So these are the parents that are taken for the cross. When hemophilic male marries a normal female, what will be the offsprings that will be produced? So let's see the genotype for this. The genotypic chart for color blindness as well as hemophilia are exactly same to same. Only thing is, instead of this C alphabet, we have to write alphabet H. H will represent hemophilia. So wherever we have written C, instead of that we will be writing H. So the genotypic chart for hemophilic male and female, it is like this. This is normal male, hemophilic male, carrier, males are generally not carriers. This is normal female, this is hemophilic female, and this is a carrier female. So it is exactly same to color blindness. Only thing is, instead of the alphabet C, we will be using H for this. So the genotype for hemophilic male, it will be X, and for recessive gene we will be using small h, and males they have X and Y chromosome, and the gene will be present only on X chromosome. Normal female, this will have both the genes as dominant. So X capital H, X capital H. So these are the genotype for male and female. Gametes, their gametes will be, it is X, small h and Y chromosome. They both are same, so they will produce similar gametes. It will be X, capital H, X and capital H. The next process is fusion of male and female gametes. So this we have to cross with this one. So it will become X small h and X capital H. But as per rule, what we have to write first? X capital H we have to write, and then only we'll be writing X small h. Similarly, this we have to cross with the other one. Again, this is also same. It will also become X capital H, and this is X small h. So X capital H and small h, both these are carriers. So both the daughters, they will carry the gene for hemophilia, hence they will be carrier daughters. Now we have to cross this male gamete with the female gametes. So this is Y chromosome, this is X chromosome. So on X, dominant gene is present, and this is Y chromosome. Dominant means this is normal, son. Again we have to cross this Y with this one. So again it will become what? This is Y, this is XH, but our rule is first we have to write X chromosome and then only we'll be writing Y chromosome. So this is Y chromosome. When dominant genes are present, that means they are normal. So this is also a normal son. So from father, the genes are transmitted to the daughters, due to which the daughters, they will be carriers. They will carry the gene for that particular disease, but they will not suffer from the disease. Now, when this carrier daughter, it marries to a normal male, now what will happen? Now the parents for the next generation will be a carrier daughter when she marries to a normal male. These are the parents. Their genotype will be, see the genotype for carrier daughter, one has to be capital and the other has to be small. So this is X capital H, this is X small h. For normal it will be X capital H, and on Y there are no genes. So these are the genotypes. Then comes gametes. Their gametes will be, they will produce similar gametes. So this will be X capital H, this will also be, this will be X small h. Since both are different, one is dominant and the other one is recessive, they will produce two different types of gametes. One X chromosome will carry dominant gene and the other X chromosome will carry recessive gene. Whereas in this case, on the X chromosome, dominant gene is present. There is no alternative allele on Y chromosome. Then fusion of gametes, this X, it will fuse with this one. See, on this X chromosome dominant is present, and on this also dominant is present. So it will represent X capital H, which means this is normal daughter. So this is normal daughter that will be produced. Then this we have to cross with this one. It will become X capital H and Y. X capital H and Y. This represents normal son. Now we have to take this gamete, male gamete, this female gamete, and then we have to cross with this male gamete. So now this is X on which recessive gene is present. This is the X on which dominant gene is present. So first we'll be writing dominant gene and then only we'll be writing recessive gene. When both the genes are present, that means dominant as well as recessive, that means it is a carrier daughter. Now this one is remaining. So this we have to cross with this one. This is X carrying recessive gene and this is Y chromosome. So this is X carrying recessive gene. This is Y chromosome. So when recessive gene is present on X chromosome and the sex chromosomes are X and Y chromosome, then this is a hemophilic son. That means this son will suffer from the disease that is hemophilia. So in the first case, the genes were transmitted from father to daughter. When the daughter grows and marries, she transmits the genes to her son. So this type of inheritance, it is called as crisscross inheritance.